Okay, now we are ready to start, and this morning's session will start with Professor Simon Laughlin. Simon is Emeritus Professor of Neurobiology in the Department of Zoology at the University of Cambridge and a Fellow of the Royal Society. And Professor Laughlin has discovered design principles that apply to many brains because they are deeply rooted in biophysics, cell biology, and information theory. His book that I would recommend very much, Principles of Neural Design, by MIT Press, co-authored with Peter Stirling, was awarded several awards and mention for the quality of the writing, other than for the quality and the importance of the topics covered by the book. So the title of the talk of Professor Laughlin is The Neuronal Advantages, Capability, Adaptability, Evolvability. Thank you, Simon. The floor is yours.
So, thank you for inviting me. It's always very good for me to come to meetings and workshops on cognition because I really don't know what cognition is. And so I'm still hoping to learn. As you've heard, I'm the neurobiologist, and I work on something which actually must be very important for cognition. It's at the root of cognition, and that is the ability of neurons to process information. And I've studied the ways in which neurons are adapted to work efficiently and effectively within biophysical and cell biological constraints. And in the last couple of years, I've realized that many of these things that I know make neurons very capable make them also, and make them highly adaptable, which is something I've worked on as well, also help promote the astonishing evolution of nervous systems, which over the course of 500 million years have gone from very, very simple structures to the things that we have in our heads today. And so what I hope to tell you is how capability, adaptability, and evolvability of how capable, adaptable, and evolvable neural systems are, and how this might help us understand the rapidity and the complexity of which nervous systems have evolved and the complexity that they've managed to achieve. So, information is essential for life, and much of the information used by cells is processed in circuits. And this shows two circuits, one the internal chemical circuits of E. coli, and the other a neural circuit. And these circuits share something in common. They're made up of a number of elements, which in sort of mathematical theory of networks are called nodes, and each node receives one or more signals from other nodes, and those connections are specific. And then the node uses these inputs to formulate an output and transmits that specifically to other nodes. And what the node does is it changes the signal when it processes it so that the input to the node doesn't correspond to the output. In other words, information is processed. And this is how circuits work. The ones in your phones, the ones in your TV sets, and so on, the ones inside bacteria, and the ones in brains. They all share this in common. Now, the thing is, the problem is that in the E. coli circuit, a lot of most of the signals are carried by internal chemical messengers, which diffuse within E. coli. Oh, I should just say that for those of you, why I'm confused about cognition is that, as Dennis Bray pointed out in his book, Wetware, these internal circuits in bacteria support cognition. They support the elements of control, resource allocation, pattern recognition, evaluation, decision-making, pattern generation, and memory. So anyway, where does cognition begin? So multicellular organisms, the chemical circuits are too slow over longer distances, that's about 10 microns, to be able to coordinate activity over the longer distances that you have in a multicellular animal. And the solution to this constraint has actually been discovered by protozoa in, for example, paramecium, and that is to use electrical signals. Because electrical signals travel extremely rapidly, they can be specific, and they can be used to drive behavior by, for example, opening calcium channels, which will activate biochemical pathways within the cell. And I'm not going to go into the details of how this works. You can look in our book if you want to. But there's a big constraint on paramecium for electrical signaling, and that is it's only got one membrane. And the membrane is the link, the connection, that's actually linking all the nodes in this circuit together. And if you've only got one connection, you can't really process very much information. And nervous systems have got around this by devoting cells to the function of a circuit. There are cells, neurons, which form nodes and form connections. And the nodes integrate synaptic inputs electrically, and the signals are transmitted electrically between the nodes. Now, there are other forms of transmission as well, but the ones which solve the problem of how to integrate large amounts of information quickly and distribute them over a larger brain or a larger body, that's done electrically. And so what we're now going to look at is, first of all, we'll look at electrical transmission and how that might have contributed to the adaptability of brains. So this is transmission measured in a dendrite of a cortical pyramidal cell, and at the top of the dendrite, an EPSP is generated, an electrical potential, and it travels 400 microns to the cell body. And as it travels to the cell body, its amplitude is reduced, its peak is delayed, and it becomes wider. It's been smoothed out in time. And the diagram on the right shows you that this smoothing is very simple from the simple biophysics of the dendrite. It's from cable theory. And so transmitting signals passively attenuates them, slows them down, and it makes it into a low-pass filter. And you can actually see that a very slow signal, that's the dotted line on the right-hand side, passes down the cable almost completely intact. So this enables you to separate fast and slow signals, which is very useful. The problem with this is that the transmission range is less than a millimeter for fast signals and less than 5 centimeters for slow signals. And you all know the solution to this that's evolved. It's the action potential. Because it regenerates, it travels long distances, and it's less sensitive to noise, but in fact it transmits information at lower rates than the dendrites, which is why the dendrites do a lot of the computation in the brain, because they can handle large quantities of information. And the reason that the action potential has a low information rate is that there are gaps between the action potentials, and during those gaps, you don't know what's happening until you get the next action potential. Now, the mode of transmission that's adopted by the neuron depends on the way the ion channels that it places in the membrane. And you all know that in order to get an action potential, you have to have the right combination of sodium and potassium channels, and that's specified by the computer in every living cell. You also know that myelination increases the speed and the economy of axon transmission, transmission along action potentials, and that tells us that glia also play a very important role in the evolution of more capable nervous systems. So what's interesting, a very elementary observation, which really is at the root of everything I have to say, is that as we've seen, signal transmission provides opportunities for information processing. We've seen that the dendrite, passive dendrite, can be a frequency filter. The action potential marks events with very precise timing. If you send it down an axon, there's a conduction velocity, so there's a delay, and in the brain, those delays are used to compute things like differences of timing in the ear. It's used for decision-making all over the brain. You collect the inputs, you weight them, and if they're statistically significant, an action potential will fire. And finally, it eliminates noise. So here is a very important general principle. Every mechanism in the brain offers opportunities for information processing and adaptation. A mechanism changes the signal it transfers. That transformation processes information. The changes are specified by the mechanism's transfer function. And the mechanism's transfer function is determined by the biochemistry and biophysics of the neuron. And that is specified by the computer in every living cell. So, and in fact, most mechanisms offer opportunities for improvements in information processing and for adaptation. And one of the themes of neuroscience in the last 10 years is the increasingly realization that almost everything in the cell is adaptable and changeable due to having the computer in every living cell. So it's not like the cartoon neurons that you see in so many psychology textbooks. Now, another property of electrical signaling is one which people have by and large taken for granted, but I think it's really, really important. And that is electrical signaling allows you to integrate simultaneously hundreds of inputs at a single node. And I defy you to design a chemical circuit that is able to do that. And the reason the electrical circuit is able to do this is because it's using the flow of current through resistances. So here is the cell body, a model of the cell body and dendrite of a motor neuron, and it's collecting with its dendrites synaptic inputs over a field of about 250 microns. And it's a remarkable feature of one of these electrical circuits is that if you change the input of any one of those synapses across that dendritic field, the voltage of the neuron will go to a new value. The time it takes is dependent by the biophysics and the cable properties and so on, but it does this many-to-one integration very quickly. And that is extremely important. Now, this electrical integration has a transfer function. This is the circuit diagram that many of you learnt about in your first year in neuroscience, and many of you would probably quite happily like to forget about it too. And it shows the circuit is very simple. You have two opposing sets of channels, sodium channels producing inward current and potassium channels taking outward current. And your input in this simple example opens some sodium channels, and the potassium conductance is kept constant. And you measure the voltage of the output of this very simple circuit, and the relationship between the input and the voltage output, the input is the number of open sodium channels, is plotted at the bottom. And it follows this familiar saturating relationship. Now, this already shows that, in fact, in this linear range at the bottom, we're having addition, and if we in fact tweak a few potassium channels, we can have subtraction as well. Also, if you plot this curve, you plot the response versus the log of the number of open channels, you'll see that the curve is a straight line through much of its operating range. So in this region, this simple circuit has taken the logarithm of the input, and this is widely used by sensory receptors, and it's at the foundation of the 𝗦𝗶𝗺𝗵𝗹𝗶 law. Now, if you open some potassium channels, you shunt the sodium channel input, and you get a smaller response, and these logarithmic curves shift to the right, and that's equivalent to division. So even the simplest, most elementary mechanism provides opportunities for computation, which are widely used in the brain. Now, of course, these resistances that are driving the integrating neuron, they have to be changed in value by the inputs to the neuron. And these changes, as you all know, take place at synapses. And synapses have a number of advantages. First of all, they're very small and compact. Second, because they're compact and the gap between pre- and postsynaptic neurons is small and the structure is small, they're very fast. They're also very powerful. A single action potential coming into one synapse can activate hundreds of channels on the postsynaptic membrane. So they've got built-in amplification, which also means, of course, they've got gains that you can change. So that makes them highly adaptable. And finally, as we'll see later in the talk, if I manage to get there, they're modular. And these are all factors which have promoted the evolution of very efficient and effective nervous systems. So one thing that synapses do is that they produce postsynaptic potentials with different waveforms: IPSPs, EPSPs, long potentials, short potentials. The different polarities allow you to do addition and subtraction, that you can change the gain of the synapse, so you can change the weight of the synaptic input, and the different waveforms give you different computational primitives for analog computation. So they're very powerful for analog computation, and it's analog computation which is responsible for much of the changes of the signal from the input to the output of the node. Also, the longer duration of longer lasting synaptic waveforms give you the opportunity to process signals over time and establish relationships over time, because with a long-lasting signal, you can use that as a kind of very short memory of what's gone before, and that allows you to compare what's happening now with what's just happened. And this can be very easily achieved by simply manipulating the synaptic mechanism. And this is my most complicated slide, but it's worth, talk like this, you can think, tell you all these wonderful things, it's all bullshit, right? So it's good to have a little bit of hard fact. And this is from a very nice review by 𝗦𝗶𝗵𝗹𝗵𝗵 and 𝗦𝗵𝗵𝗵𝗵. And there are two sorts of synapse, at least two sorts of synapse, which in mammalian nervous system, which use excitatory and use glutamate. And their types are named after the drugs that inhibit the postsynaptic receptors. The AMPA receptor has a very fast response. It only lasts time constant of less than a millisecond. And this is achieved by packing the receptors, postsynaptic receptors, very tightly on the membrane directly in front of the release site. So when that transmitter is released, it hits all of the receptors almost at the same time. And they rapidly saturate it, so it rises to a peak very quickly. The receptors have their biochemistry adjusted so that they have a very low affinity for glutamate. They don't bind it tightly. So the glutamate is almost immediately released and diffuses away from this small cluster. So the concentration of glutamate crashes down really quickly in the cleft and terminates the PSP. And the glutamate is quickly mopped up by glia transporters, which are placed close to the synapse and are very powerful and have very high affinity, and they're there in high density. So this is molecular engineering involving collaboration between neurons, pre- and postsynaptic neurons, and glia, all to achieve a very fast response. The NMDA receptor does the opposite. The receptors are scattered over a large patch, so glutamate release takes some time to get to them, so it takes time to rise. It has a very high affinity for glutamate, so it hangs on to it for a long time. So it has a very long memory, if you like. And that's actually the basis of how the NMDA receptor supports memory. And the glia transporters are far from the release site and at rather low density. So the glutamate hangs around for a long time. And you can see that the opportunity to engineer these two synapses using the same transmitter to have very different responses comes from the ability of the computer in every living cell to select exactly which receptors will be expressed on the membranes and specify the structures in which they're expressed. Now, the synapse is often depicted as something very simple, but in fact, we are becoming increasingly aware that the synapse is really a microprocessor. That below, particularly in the postsynaptic membrane, there are very complicated biochemical networks which can achieve all sorts of changes in the postsynaptic cell and can also produce long-lasting responses by feeding back and activating channels. And we now know, for example, that in the synapse, in mouse synapse, there are something like 1,800 distinct proteins, either isoforms or different protein genes being expressed in the mouse brain. And that each one of these, each synapse probably uses about 180. So there's an incredible amount of diversity and molecular engineering in synapses. Because it's part of the wetware of the computer in every living cell, when information comes into the synapse, it can be processed by the synaptic microprocessor, which is part of the computer in the living cell, which can then rebuild anything virtually in the cell. So we have the most astonishing prospects for plasticity. Now, what about making the connections? Making sure that the synapses connect to the right nodes. This is essential. Intracellular circuits use binding specificity largely between proteins. These could be either mechanical or chemical binding. And this means that if you have nodes using the same chemical messenger, they have to be far away from each other. Otherwise, their signals will interfere. So if you want to pack nodes very tightly and have them being independent, then you can't do that very much because there are very few of these chemical messengers available. Electrical transmission via dendrites and axons eliminates this chemical crosstalk and allows very dense interconnectivity. And this little diagram here shows a few cubic microns of the neuropil of Drosophila. And that's made possible by the use of axons and dendrites. So the complexity of the wiring of the circuit can be greatly increased. Now, they also eliminate another constraint, which is the number of independent connections that can be made in a small volume. If you only have, in your intracellular circuit, if you have n species of messenger molecule available to you, you can only make n connections in a small volume being a few microns, and n being of the order of 30 or 40. Because the neural circuit connects using mechanisms that direct growth, it has many, many more degrees of freedom for making connections than chemical circuits. In addition, the synapses localize chemical transmission to eliminate crosstalk, and the number of connections that can be made onto a single node in the neuron is only limited by the surface area of its dendrites and the footprint, the size of the area of contacting synapses. So a single neuron can make very large number of independent connections, and this Purkinje cell makes something like 180,000. And you simply can't do that using chemical circuits in a bacterium. So the story so far: neural circuits enhance processing speed and computational power by overcoming constraints that operate in intracellular circuits. The computer in every living cell can draw from a very large part list of available signaling molecules and assemble them in many different ways. And it can nano-engineer these systems to make sure exactly the right channels are expressed in exactly the right place at the right time. So we have an enormous design space for constructing circuits. But within this design space, only a tiny fraction will work at all, and even fewer will work well. So the successful adaptation of circuits to function depends on the ability of neurons and glia and the cells associated with their development to navigate this design space and settle on some good places to be. And this brings us to adaptation and adaptability. And this is a tale of two timescales. There's adaptation within an animal's lifetime, and then there's evolutionary adaptation over very long timescales. And we'll start with adaptation over an animal's lifetime. And here's a really nice example of phenotypic plasticity, which comes from a friend of mine, 𝗦𝗶𝗺rie, who has the lab across the road from me in zoology. When the population of density of locusts is very low, they avoid each other so that they can maximize their foraging and finding good places to live. But once the population density arises, locusts start bumping into each other, and they start seeing and smelling other locusts. And an individual locust switches from being repelled and staying away from locusts to approaching locusts and being close to it. And Malcolm Burrows and his group showed that all you need do is just brush the bristles on the hind leg of a locust with a paintbrush for a few seconds, and it will switch from solitary to gregarious. And that this switch is achieved by the neuromodulator serotonin, which in fact reconfigures the circuits in its brain to change its behavior. So, looking more closely at this within lifetime plasticity, the changes occur on a well-established nervous system. And the neurons and the circuits are responding to context. So they're responding to two types of context: the correlations within or between neuronal signals within the nervous system, so what your neighbors and people nearby are doing or not even nearby, and as we've seen in the phenotypic plasticity, the changes in this animal's status and environment. And of course, all of these changes depend on the computer in every living cell to actually make the changes in the brain that causes this stuff to happen. So let's look at, there's some very obvious examples. I'm not going to dwell on this for a great deal of time. Correlations between the signals within and between neurons. If a neuron is powerfully stimulated, and that powerful stimulation remains constant, so the signal is strong, there's a strong correlation between the signal I'm getting now and the signal I just got recently, it's the same, the neuron adapts, as shown by Adrian and Zottman in 1926. And there are other examples: synaptic depression and facilitation. And this adaptation removes redundancy and adjusts the gain and so on. So that's adaptation to context within your own signal. Then there's another form which has been discovered more recently, which has been called homeostasis. I'm not quite sure that's the right term. You can discuss that with me later. This is adjusting the sensitivities of the membranes of neurons so that they maintain, the circuits keep in balance. So if inhibition or excitation are too strong, a neuron will either fire too little or too much, and that's going to be a disaster. And all of these circuits only really work well if the inhibition and the excitation are kept at the right sorts of level. And so if you take a neuron and you starve it of action potentials, you apply a drug which limits the number of synaptic inputs coming to it, It responds by reducing inhibition, reducing the number of potassium channels in its membrane to lower its threshold, and increasing the number of sodium channels. And so this is responding to the inputs that you're getting from other cells to make sure that you have the right sort of gain. And finally, there's a context-dependent change in synapses, which you all know about, and this is spike timing-dependent plasticity, in which the relationship between two signals in the neuron, which are the EPSP and then a spike, depends on the temporal relationship between the EPSP and the spike. If the EPSP occurred before the spike, so can be causal, then that synapse is strengthened. If it occurs afterwards, then it's weakened. So this, which is often regarded as a sort of phenomenon, all in a class of its own, is just one of many, many examples of context-dependent adaptation. Changes in an animal's status and environment, these are so widespread and so wide known that really I'm not going to talk about them at all. I want to get on now, at the end, to the evolutionary factors, the factors that might change the ability of the nervous system to evolve, to explore and to expand, but also to contract the design space. So, the way in which dendrites, axons, and synapses connect up promotes the evolution of new connections. As I've already said, there are many, control of growth has many degrees of freedom. So there are many mechanisms involved which can be targets for selection and adaptation. And just to, we can see why there are targets. Growth cones actively explore to find their contacts, and neurons seeking contacts will actually compete to make contact. So these are not genetically specified. There are lots and lots of behavioral processes going on determining how the connections are made. They're not rigid programs. In addition, they have the property that they are able to actively correct for errors. So they are goal-directed processes which have some feedback which enables them to get to the right place even though they might have made a small error earlier on. And if you don't believe me, you just have to look at some neurons. So these are 3 identified unique neurons in a cockroach. A cockroach has one of each of these neurons, and each of them is doing a slightly different thing in the auditory system. And people looked at these 3 neurons in 3 different cockroaches, and they filled them up with dye. And so for these 3 types of neuron, functionally identical, individual, identifiable neurons had slightly different dendritic trees. The general form and location is the same, but the fine branches are all different. And that's symptomatic of a system which uses growth rules to home in on a final target. And what that means is that neurons are sending out dendrites to places that they're not meant to go to. And that gives you the opportunity to make new connections. A second property which promotes evolution, which has been speculated about quite a bit, is that synapses are modular. And this is the definition of a module in evolutionary biology. It's a system, a self-contained system, with lots and lots of interacting mechanisms, but very few inputs and very few outputs. That's a synapse. The interacting mechanisms provide many, many opportunities to change that transfer function, to change what the module is actually doing. But you can make those changes without disrupting the inputs and outputs. And you can do it independently in different types of synapses. So modularity is a big plus. Synapses also solve some very practical problems, and I don't think this has been drawn attention to at all, some very practical problems to do with power supply in the brain. So if you design a circuit, you have to make sure that every component in the circuit is attached somehow to the power supply or to another active component. Now, in a synapse, there's no direct electrical contact between the presynaptic neuron and the postsynaptic neuron. So current, that when an action potential arrives at a chemical synapse, current is not driven into the postsynaptic membrane. That means you can attach lots and lots of synapses to a neuron without draining all its current, and you don't have any impedance matching problems. And that actually gives you a great deal of freedom to make new connections. In addition, cell biology takes care of the energy supply. Every neuron contains its own mitochondria to make its own energy. It can control the size of that population of mitochondria, and current estimates are that 40% of these mitochondria are mobile, and they're able to go to the sites where demand is highest. So you plug your synapse in, and everything else, electrical problems, they're all taken care of. So the final thing I want to talk about is efficiency. Efficiency, I think, is important, and I think there's one paper by Jeremy Niven which has drawn attention to this. Efficiency is very important because it gives you a reason to finely engineer and adapt circuits, which is not to do with making more advanced or better behavior. It's to do with doing the thing that you're already doing a little bit better, more efficiently. And Peter and I, in our book, we came up with 10 principles of neural design, and many of these will promote evolution. One of our principles, compute with chemistry, because it's cheaper. That's use the computer in every living cell. One of our principles is that every neuron should minimize the amount of information and the number of action potentials it sends to the very bare minimum necessary to do the job. And for that regulation of sensitivity, you need very accurate homeostasis. We also say that you should only send what is needed, not noise and not redundancy, but signal. And that will promote the evolution of filtering and adaptation and lateral inhibition and predictive coding without there being any overt behavioral need for these mechanisms. I think match, adapt, and learn, that's pretty obvious. Another is to make neural components irreducibly small. And you see lots of examples of that. And that's going to really sharpen up the computer in every living cell to be as accurate and precise at specifying things as possible. And it should also control the morphology very accurately to minimize the wiring in connections. And a lovely example of this has come out recently. This is a stretch receptor neuron in the body of Drosophila larva. And what this is plotting is the diameter of the branches. Notice that it's in nanometers, so they're very fine branches here, as a function of the leaf number. Now, the leaf number in a branching network is that if my fingers are the leaves, right, then my arm has a leaf number of 5, and my whole body has a leaf number of 10. And so it's the number of tips in the branching network that your particular node supports. And they show that the diameters of these dendrites are very precisely controlled so that they're appropriate for the number of tips that they're supporting. And in fact, cross-sectional area of the dendrites is scaling in proportion to the number of leaves. Now, another principle that we came up with is complexify, make things more complicated. And we got this idea from engineers, in fact, an engineering handbook, in the German engineering handbook on the process of design, said that it's often very inefficient to have one component trying to do two things. And the classic example for those of you who are petrol heads, who have probably nobody left in the room virtually, is the two-stroke engine versus the four-stroke engine. So you add a new component so that each component can now do its task as efficiently as possible. And the design handbook says this is beneficial because it offers you starting points for new designs you'd never thought of before. So making things more complicated, people in neuroscience and biology in general like to think of Occam's razor because that helps us understand it. But in fact, biology is setting out to make things more complicated because that's going to be more efficient. And this means that another way in which efficiency is going to drive control and adaptability and complexity. And there's little doubt that what I've told you about today supports cognition, better cognition, because better. Because it's generally agreed that there are two changes which have to be made to make nervous systems more complicated and able to process information in more elaborate and in deeper ways. The first change that you have to have is that you have to be able to have more complicated wiring so that you can bring more things together and combine them with other things in many different ways. And the second thing that you have to have is the ability to perform more complex and more penetrating computations on the inputs that you get from those connections. And that, of course, means having more complicated signaling mechanisms and more complicated circuits in your cell. And I think I've shown you today that very basic neural advantages, which stem from very simple properties of neurons and neural circuits, actually provide the opportunities to produce the very complicated brains that we see today. And what we don't know is what the limits to this type of complexity really are. Thank you.
Thank you very much, Simon, for this scholar and wonderful lecture. Is there any question? Massimo, give you the...
Thank you very much for the wonderful talk. I wanted to ask you something. Listening to this story about the neuronal complexity made me realize something that maybe I knew, but I never considered this way. That really the design space that neurons have is linked to their imperfection. It's about the presence of noise, it's about the individual differences between neurons, it's about how the transmission works. You know, we had the occasion to invent things because the system was in some way had space for changing and for imperfections. When we make engineering systems, we start from a completely deterministic approach. Like it's 1 and zeros, and then we spend a lot of time adding components to mimic what actually the noise left us able to do. Do you think that we are kind of losing the most important part when we try to remove noise from our systems? Or, I don't know, what's your opinion on this?
So first of all, your observation that the evolution of neural circuits is partly driven by noise, that's very profound. And I completely agree. And I think 𝗏𝗏 said this, and I've said this. If there was no noise in the brain, then every signal, no matter how small, would be statistically significant. So it wouldn't matter how you process the information, it would still all be there. But of course, that's a fantasy. It's not a fantasy in digital systems because they've been engineered to get away from noise, and in doing so, they also get away from problems of impedance matching and so on that I've talked about. But nervous systems are not digital systems. They're mixed, they're hybrid systems, and I don't like to call them digital because I don't think they're Boolean most of the time. There will be bits which are Boolean, because that must be advantages to that. But most of the time, they're mixed analog and pulsatile. And the analog processing is much, much more efficient energetically than the pulsatile processing. It's about, analog transmission is about 12 times more efficient, and something like a multiplication, which takes 8 steps or whatever, depending on your precision, in digital, is done directly by that simple membrane that I showed you. So analog is much more efficient. So noise has been very important in determining the way in which circuits are configured and how they've evolved. And actually, one of the things that noise introduces is that very often you need to have several synapses driving a single neuron with a single input, and that introduces some redundancy in the system, which also promotes evolvability. Now, there's a sort of coda in the end of your question, which is actually, is there not a role for noise in computation? And yes, there is, but I don't know enough about it, and we haven't got time to talk about it. Because I think there are other questions.
Thank you very much.
It's a very good question. Thank you.
I mean, thank you very much for this very concise overview of all neuroscience and neurobiology of neurons. It was very nice to see. I have just one comment on the synapse. You said that they basically have no direct connection, but they're also these electrical synapses that have these gap junctions and
yeah.
so on. So you have this impedance problem anyway.
So I mean, you're right. So electrical synapses are used, and they're used because they're very direct and very fast, and sometimes you need that. Basically, Golgi was wrong in saying that the nervous system was syncytial, and 𝗂𝗏 was right, because in Golgi's syncytial nervous system, if you fired an action potential, it would have almost disappeared completely by the time. The signal would have disappeared completely by the time it had gone through 2 or 3 layers of processing. So, yeah.
Thank you. I also enjoyed very much the talk. I have a question because on cognition, because it seems that these systems are very advanced. You mentioned the spike timing dependent plasticity, for instance. They can take into account the circuits, multiple dimensions. So then, what happens with cognition because we miss some of the computational skills that instead neurons can process, right? So is it a cost of modularity, or how do you see this mismatch, that something that is available is not available at all levels? Thank you.
So I don't quite understand, when you say that there are these things happening in cognition, you're talking about slower processes in cognition. The fact that...
For instance, the processing of time.
Yes.
You mentioned something that happens before or after, which is a computation that in some domains we struggle, many animal species struggle to take order into account, whereas at the lower level, let's say, it's straightforward and it is acquired by many neurons. So why something that is in principle available at the lower level then struggle? What is your view of these differences in layers?
Yeah, that's a very good point. I mean, first of all, I think I agree with you that I think time is the last frontier in neuroscience. It's a dimension that we actually know relatively little about how information is being processed in time. Now, in terms of the circuitry of neural circuits, in principle, there's no problem with time. For example, every cell contains a circadian clock, and that's running on a 24-hour cycle using a cyclical chemical reaction. And you can imagine there being little cyclical chemical reactions could be set up in cells with lots and lots of periods. And a very good example of the ability to process time in even a simple nervous system is in C. elegans will associate the taste of a bacterium that it's eaten with the fact that sometime later it becomes sick. Now, if you think about it, in a world full of bacteria and food poisons and so on and poisonous food and so on, this is something very, very important for an animal to know. And so this ability to associate what was in your mouth some time ago with the way you feel now is really fundamental to survival. And I think there are several examples now where people have looked at gustatory memory in C. elegans and in locusts and shown that there are short-term memories which allow you to make the animal to make this association, but of course the mechanisms as yet are unknown. So I think the wonderful computer and living cell can solve a lot of these problems. The thing is, you won't see that down the barrel of your electrode unless you can hold on to a neuron for a long time and actually discover how its membrane potential might be changing, for example.
Thank you, Simon, for this, as ever, thought-provoking talk. So I found it really inspiring and perplexing. So my question comes from the perplexing parts. So the inspiring part, I think, is that if one is to choose at which level to focus on principles, I think this is the right level because, for example, there will be no point in focusing on neural architecture principles that are specific to humans, so specific, like, cortical organization. So I think that's also where I would go. What I'm left unsatisfied by, and this goes back to what you said at the beginning, you came back to at the end, is the link with an explanatory account of cognition. So,
an explanatory account?
An explanatory account of cognition itself.
Of cognition, oh yes.
So, so if one were to follow this program to its full conclusions, one should imagine that at some point in the future we're going to have an account of cognition that starts with these principles and is able to reverberate them through all the way to behavior. And I'm just unconvinced that that's a fruitful program. And we could discuss many examples. I mean, what Massimo mentioned, for example, let's take noise, okay? Of course, we have neuronal noise. It's a fundamental aspect of our data process and so on. We also have noise in behavior. The idea that noise and behavior bears a relationship with neuronal noise that we can understand is, to me, not transparent. For example, noise and behavior could reflect exploration. It could reflect other factors. In other words, it seems to me that every time we try and explain cognition, we come back to accounts that operate on a completely different level. And so my question to you is, do you really believe that there is, at some point, there is a light at the end of the tunnel where we will be able to build this link? I just remain unconvinced, and we have evidence from many other disciplines like physics where that kind of program has failed, essentially. So that's the question. Thanks.
So will we ever completely understand the human brain? I kind of hope not. And I think there are a number of reasons why that's very, very challenging. Not the least of which is that actually to make an observation on the brain of any animal, you have to some way restrict it and interfere with it. And that actually disrupts the sorts of things that the brain is doing. So the one thing that I haven't mentioned is the link between the things that neural circuits are doing and the animal's behavior. And so if one is to be guided through all of this potential, so we're kind of searching this space as well. We're searching for the design solutions that that brains have evolved. But if you want to understand design, you have to actually have a very good understanding of function. You could not understand the design of a modern aircraft if you knew nothing about the aerodynamics of flight and what planes are able and not able to do. So it's keeping this close link to behavior is very important. But I agree, the prospect of how we actually ever understand completely what brains are doing is daunting. And you have to really start with the question, well, given that I can't understand everything, what should I be? What am I trying to understand, and why am I trying to understand it? So while the microphone's traveling, I'll just say that one of the advantages of being the first speaker is that there's lots of time to discuss things with people. So if any of you have any questions, no matter how, there's no such thing as a stupid question. There's certainly lots of stupid answers, but there's no such thing as a stupid question. So please stop me and ask me, because I know I've covered a lot of ground, and there'll be things you don't understand. I think there are things I don't understand, but I'm not going to tell you which.
If there are no other questions, I have a question, Simon. Because I was wondering if there is a way, in principle, to predict or to know in advance if some structure or mechanisms in the brain is there, as you say, to increase in some way information processing, or just for the ordinary function in a particular environment. I have an example which is related to a previous edition of this workshop. There was a colleague, a neuroanatomist, working on dolphin brains. And I mentioned that because you mentioned myelinization and the role of glia. And you know that the brains of dolphins are very large in volumetric terms, but he investigated the brains of dolphins of different species, and he found that actually the size of their brains is not related to the number of neurons, which are quite sparse, but rather to the glia. And the reason has to do with the heat control of the glia. And so he found that there is a correlation between the size of the brain and how cold is the sea in which the particular species inhabit in it.
Yeah. No, I mean, that's quite possible. So, you know, natural selection does not say, well, it says in you know, Kandau et al. That when you want to perform, that this structure is here to do that. Natural selection takes opportunities. And if glia are generating heat and the brain is cold, then they'll start making brown fat and mitochondria. And the nicest example of that is, in many ways, are fish, mesopelagic fish that are like tuna, which are very fast predators, but it's very cold. So their eye, they're cold-blooded, their eye works very slowly. So they have a special heater organ for the eye, which is in fact modified eye muscle, which contains lots of brown fat and mitochondria, and they have a special circulatory network, the 𝗦𝗂𝗵𝗹𝗶𝗵𝗵, which goes over the back of the retina to carry the heat from this heater organ to the retina.
Thank you. Is there any other question? Yes.
Thank you for your talk, first of all. I have a question about the waveforms of action potentials. So basically, let's say you are recording an extracellular action potential in a freely moving animal. And in the current time, many of my colleagues create toolboxes and pipelines which lay on the assumption that the waveform is stable over time. But I'm wondering, for example, if you have a neuron like a dopamine neuron, which is encoding a homeostatic state of hunger for a mouse, and the mouse is eating, therefore the priority that that neuron is assigning to food changes, and it also has an impact on its firing rate, amongst other things. Is it reasonable to think that there are also other systems inside the dopamine neurons which start changing, therefore which will start affecting also its waveform of the action potential?
I think that's it's certainly perfectly possible to do that. We know that action potential waveforms change just actually due to things like persistent sodium channel activation and so on. But for example, in the cockroach, the waveform of the action potential changes from summer to winter, because when you're operating at low temperature, the kinetics of the sodium channels are not appropriate for producing a fast action potential. So, yeah, it's certainly something that can be done, and something that one should think about as a possibility.
Do you think it's possible for these waveform shapes to change from like even triphasic to biphasic, or to change even over the course of 1 hour if there has been any significant event related to that neuron's function? If you have a vibe about it.
Are these triphasic because they're extracellular, or no?
Yeah.
Well, and the other thing the waveform depends on in an extracellular action potential is the extracellular resistance, so the glia could change as well. So the answer to your question is that it might not be a totally safe assumption that action potential from a single unit remains stable over time. And somebody actually ought to test that. And the obvious test would be to record from a neuron intracellularly and at the same time record its extracellular potential. And first of all, see how well they correlate.
Yeah.
And then, yeah.
So like patch clamp plus some FS implant, basically. Yeah. Okay. Thank you.
But I mean, you might not get anything. If you wanted to write a grant for this, you'd have to have some preliminary data.
Okay, if there are no other questions, please join me in saying thank you very much and a big applause to Professor Laughlin for this talk. Thank you. Now we have the first poster session and a coffee break. And the next session will be at 11:30 sharp, please. Thank you.